Hello बच्चों, in this video I am going to teach you structure of cell. So first of all, very quickly I am going to draw a cell, animal cell. So even you can draw in a circular one, but I am going to draw according to your book. So this is the cell membrane. As we know that in the case of the animal cell there is no cell wall. So this is cell membrane simply. And then next is that is nucleus. As we know that the nucleus is the largest cell organelle. So the nucleus is there in between inside the cell, and of course nucleus is surrounded by double membrane. So this is membrane 1 and this is I'm going to draw the second one. This is membrane 2, leaving some area free in between. This is to be termed as the nuclear pore, so that it is connected with the cytoplasm outside. Next, that is inside the nucleus there is the nucleoplasm. So this is the nucleoplasm, that is a fluid part. In this nucleus there is a thread-like structure, and these thread-like structure are also termed as chromatin network. So I am going to draw 4 thread-like structure, 2 and of course 3 and 4. In one of the thread at a certain tip at one place there is a rounded structure without any membrane, you can say no membrane is there, and this rounded structure is to be termed as nucleolus. So I am going to draw this one is the nucleolus. Inside the nucleus this is rounded structure attached with one of the chromatin fiber, and it is to be termed as the it is to be termed as the nucleolus. For your easy reference, now I am going to do some labeling part. This is termed as cell membrane. Another name of this one is plasma membrane. Another name is also termed as plasma lemma. Then followed by this one, this is termed as double nuclear membrane. Here I am going to write down nuclear membrane. Whenever we will be using the term membrane, it is always made up of lipid and protein. This is the membrane which is made up of lipid and protein. Inside the nucleus, a rounded structure is there. It is to be termed as nucleolus. Then this thread-like structure is termed as chromatin network. CHROMATIN, chromatin network. Alright, this chromatin network later on it will form chromosome, and you know that the function of the chromosome is to carry genetic information from one cell to the another cell, or you can say from one generation to the another generation. Now the most important part, of course, inside the cell, which is making a framework like structure, it actually originates from this nuclear membrane. So in spite of using the same color, I am going to change the little bit, but as it is developed from the same one, so definitely it is of the same composition also. This is to be termed as ER. So you can see the difference in between. It is originated from the same one, same nuclear membrane. It is originated like this and responsible for making anastomose. The meaning of anastomose is that is it is highly branched like this. It is like this one. At certain places this endoplasmic reticulum is having dotted structure. Dotted structures का मतलब rounded structure. These rounded structures are to be termed as ribosomes. Sometimes, not sometimes, we can say that it is also present in the scattered way into the cytoplasm. So this is ribosome. This red colored dots are indicating you the ribosomes. And one more thing, that at certain places this endoplasmic reticulum is not attached with the ribosome. Going to do the labeling part for this red one also. This is a ribosome, discovered by the Robinson first of all. And then this endoplasmic reticulum, the whole soul, this part, this one. In short, I am going to write ER, endoplasmic reticulum. When ribosomes are attached with the ER, this kind of ER is to be termed as rough, ROUGH, rough. What is it is? ER, endoplasmic reticulum. When Ribosomes are not attached with this endoplasmic reticulum. In that condition, we use the term smooth, this one, smooth, 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 smooth endoplasmic reticulum. Now, the fluid part which is there inside this cell, it is the fluid part. So, when we exclude the nucleus, that means we are going to remove the nucleus, not from the cell, but from the definition. In that condition, for this one, we use the term cytoplasm. It means when we include the nucleus, it is to be termed as protoplasm. As the next cell organelle, as it is the animal cell, I am going to write down at the bottom also that right now we are talking about the animal cell. Alright. Now, in this animal cell, ER we have completed endoplasmic reticulum. You know, when the small portion of the ER is detached from this place, from here or from here, then ultimately it will develop into a Golgi body, and Golgi body is, the way of making of the Golgi body is that the concave surface of the cisternae, what is cisternae? A sac like structure, is facing towards the outer side. That means this surface is facing towards the outer side, and some rounded structures are also there very close to this one. They are also termed as vesicle. So, commonly this one is termed as Golgi, Golgi, Golgi body. This is your term. In animal cell, the largest cell organelle is the nucleus, and the second place is given to mitochondria. So, what is mitochondria? Mitochondria is double membrane bounded structure, and the inner membrane is responsible for cristae. I will make it separately later on after completing this diagram. One more cell organelle is present, and it is simple membrane bounded structure present into the cytoplasm, and they are termed as the lysosomes. This one is to be termed as lysosomes. The main function of lysosomes, I am going to write down SOMES, lysosomes. And this one, this pinkish one, is to be termed as mitochondria. This is to be termed as mitochondria. Right? This is mitochondria. Now, going to tell you the function of all cell organelles one by one. The function of cell organelle, cell membrane, is to separate the cytoplasm from the external environment. Nuclear membrane, to protect the inner component, internal component present inside the nucleus, that is nucleolus and of course the chromatin thread. Chromatin thread or chromatin network, later on it forms chromosome. So, it is a genetic material, it contains the genetic material. Then followed by the mitochondria. I am going to take out the mitochondria outside like this one. Now, when we cut the transverse section of the mitochondria, meaning that we are going to cut it like this one. What is this mitochondria? This mitochondria is actually made up of two membranes. Internally, it is visible with two membranes. That means membrane number 1 and membrane number 2 will show you a cristae. Inner enfoldings are finger like projection, and it is termed as cristae, like this one. On this cristae, there are some pinhead like structures are present. These pinhead like structures are termed as oxysomes or the F1 particles. These are present like this one, and inside this mitochondria there is a fluid, and this fluid is to be termed as the matrix. It is present inside this inside this mitochondria. When we talk about the membrane, that means whenever the membrane is present in around any cell organelle or of course around the cell, then in that condition, like this is the cell membrane, this one, nuclear membrane, this one, endoplasmic reticulum is also made up of membrane, this one, Golgi body made up of membrane, this one, mitochondria made up of double membrane. Golgi body itself is also surrounded by the two membranes. Nucleus is also surrounded by the two membranes. So, whenever we use the term membrane, it is always made up of lipid plus protein. So, this is actually the chemical composition of this one. Main function of the mitochondria is aerobic respiration, AEROBIC, aerobic respiration, right? In this process, any cell makes ATP. The full form of this ATP is adenosine triphosphate. You know, this ATP is the energy currency of the cell. It gives the energy. So this is the only reason why this ATP is to be termed as, because it is helpful for maintaining all the metabolic activities of the cell. Now, one cell organelle which is being left, that is present very close to the animal cell. I am going to change the color again, and this is to be termed as very close to the nucleus. So, I am going to draw it at this place like this one. It is termed as centrosome. This centrosome is also a type of cell organelle present only in animal cell. Now, listen very carefully in this diagram. There are certain components, there are certain cell organelles which are there only into the animal cell. The first one is to be termed as the centrosome. So, this is centrosome, CENTROSOME, centrosome. This centrosome helps in cell division. This is the only function, and there are so many sentences given in your book also that it helps in spindle formation, but the better way is to learn that it helps in cell division. We will discuss it later on in the next class also, in class 10th also, that what is the spindle fiber. And then followed by lysosome, they help in this one. This is also there only into the animal cell. So, the lysosomes are also there for the intracellular digestion, and also the one adjective is to be added termed as the suicidal bag. So, these are the two specific characters present only into the animal cell, and you can observe that I have not made plastid over here, right. The plastid is not to be made over here, and cell wall is absent. In that condition, these two components are there into the plant cell only. So, I am going to show you that what is the difference in that plant cell.